Okay, so let's talk about, and this one gets a little bit more involved, but let's talk about the transport mechanisms where it does need some sort of assistance, because it's going kind of against the gradient. So it usually involves some carrier proteins. As I mentioned before, there's got to be, this will be able to get in here, but this shape won't be able to get in here. So they're very specific. All right? So we have to have like a lock and key. Competition. You have two students here. You have two students that need to come into the room. All right? One of them is, one needs to go for biology, the other one needs to go to chemistry. This is a biology course. So think of this person is able to fit in here if they go into biology, because that's what's on their curriculum. The person in chemistry don't need to go in here. I'm not going to fit in here. I'll just go someplace else. So it's very specific. Competition, well, let's say there's 3 students that need to get through that doorway. You're all going to biology. You can't fit all in there. So you're going to compete for the doorway. You're going to push them out and fight to get, you know what I'm saying? All right, I see this a lot in New York City when we get the subways. All right, so there's competition going on, okay? If they have a similar shape. And saturation. Well, if we have 2 doorways over here, and there's 50 people out there, you can only have one person at a time going there. So how many people at a time can we get into the room? 2. So it becomes a saturation. In other words, if we had, let's say, 5 doors out there, and I had 5 people out there, everybody could come in at the same time. But if we saturate outside there and have 50 people with 5 doors, we can only fit 5 people at a time. Does that make sense? That's what I mean by saturated. It can be overly saturated. And the saturation is not going to change the rate of the people coming into the room, because we only have 5 doors. It'll stay constant. All right? So this is just, again, showing you if there's 2 molecules here, 2 doorways, it doesn't matter. If there's 2 molecules that need to get in, it'll work out fine. But if you saturate outside there, you can only take 2 at a time. Okay? All right, so let's first talk about facilitated diffusion as one of our mediated assisted transport mechanisms. This is going higher to lower. That's why we call it diffusion. There's a little trick to this one. Okay? It must be attached to a carrier protein. All right? So a polar molecule may have trouble passing through a cell membrane unless it's attached to another molecule that can pass through here. A little baby is not allowed in here unless it's accompanied by its parent. So the baby can't get in here. Not allowed to come in here. But if the parent picks up the baby and walks in, there's no problem that way. Does that make sense? Okay? As long, those babies could come in here as long as we have enough parents out there that can carry them in here. All right? No energy is really needed to do this. The baby actually is not using any energy. It's using someone else's, right? But that's, I don't want to get into that one. But the baby's not using energy to get in here. Okay? It may release energy. The baby may scream and release energy. I don't want to go in there, right? That kind of thing, right? But it may or may not cry. Okay? So, again, this molecule can go in here as long as it fits into this receptor, and then it pushes it out here. So it has to fit like a lock and key. So you could have 50 balls out there and 2 balls in here, as long as there's something that's going to fit like a lock and key, it'll come in. And it goes from a high concentration to a low. All right. Other things is just showing you it's a receptor mediated. As long as this piece comes in here, it will open up this. Basically, as long as I can come in here and lock the door, people can come in here. But you can't come in unless I unlock the door. And that's what it's showing there. Okay, questions? Okay. All right, so let's talk about active transport. Now, this in parentheses is really called primary active transport. No one really calls it primary active transport. When we say active transport, it's referred to as the primary. We just don't say it. The reason why I'm mentioning this is because there's something called secondary active transport, and I don't want you to, I want you to understand the concept of that. All right, but we don't usually say primary. So this is what happens. This is going to go against the gradient. Okay? This is going to go from a low concentration to a high. The opposite of diffusion. So let's get back to my situation of me flatulating and making it really strong over here. Potent smell. Right? Over here, they're not going to really like it. But Ashlyn over there is pretty brilliant. She can do something because right now she's in an area that is low concentrated with that odor. Here it's a very high concentration. She wants to make sure that the air from a low concentration goes to a high concentration. The opposite. And that's not natural. But she's pretty brilliant. Without her getting up, she is nailed to that seat. What's something she can do that can try and get the smell to stay here? Yeah, you're already doing that, right? What is it? This, right? Or you could plug in a fan, and you're going to blow the air from a low concentration to a high concentration. But you have to plug it in. You have to use energy to do that. You're doing this. You need to use energy to do that. It's not a natural thing. That's what active transport is. From a low concentration to a high concentration, but you're using ATP. You're using energy to do that. It has to be required to do that. Is that clear? Okay? So the kidney, you'll see that a lot. We're going to also have something called a sodium-potassium ATPase pump. Do we have more sodium outside the cell or inside the cell? Outside the cell. Bless you. But there's going to be sodium coming inside the cell because that's diffusion. But we have to put it back out there. We're going to go from a low concentration to a high concentration of sodium. Does that make sense? Likewise, potassium. Is that higher inside the cell or potassium higher outside the cell? Inside, right? The salty banana. Right? So that naturally, the potassium wants, as long as those potassium gates are open, the potassium wants it to rush out of the cell. We got to put it back into the cell. It's got to go from a low concentration to a high concentration. That's not natural. So we can do that through active transport, but it's going to use energy. It needs ATP. We call this pump the ATPase pump, or the sodium-potassium pump. Who here has heard of the sodium-potassium pump? All right, about half. Okay, good. So you have going from low concentration to high concentration. Energy has to be used because it's not a natural thing. So you have the sodium-potassium pump, the classic one to do this. All right? It's also known as the ATPase pump. We also have a proton pump to push protons, hydrogen ions, out there. That we're going to talk about when we get into how to make ATP, something called the electron transport chain. So it's going to come back to haunt you, okay? So the sodium-potassium pump, all right? Very important that we're going to be talking about for animal cells, because we have to get that, again, the membrane potential back to where it was. So we're going to be talking about that on and off with our biology courses, okay? And it's seen in places like the neuron, the kidney, ATP is required. The way it works is that 3 sodium go out of the cell and 2 potassium go into the cell. Remember, it's going against the gradient. There's more potassium inside than there is outside, but this is going to go against the gradient from a low concentration to a high. And you should know what these, the 3 and 2. Why, let me ask you this, why wouldn't it be 2 sodium out and 2 potassium in? Why is it so important that it's got to be off center like that? What happens to the charge if it was 2 and 2? There would be no change. 2 positive going in, 2 positive going out. The charge is going to be the same. Not good. So this allows it to go on. So I came up with a mnemonic to help you to know which one goes which way and how many there are. You ever hear that song, na 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 na, hey, 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 right? You've heard of that, right? So this is, I was doing this in college, and that song was playing, and it just hit me when I was studying this thing. Now let's listen to this. Na, na, how do you spell na? And hey, this is already, it's leading to something, right? So then I went, na, na, na. Now how many times did I say that? Look at how, somehow I think that the song actually came from this ATPase pump. Right? So you put it all together. Na, 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 out. Na, 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 out. KK goes inside. It looks like a commercial. I don't have much of a social life. All right? But if that works, memorize it. It'll help you later on. When you get into other courses, you'll learn more about the membrane potential. And the Salty Banana, the Na 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 song is going to help you. You're going to see these other classmates of yours, they're like trying to remember this and this, and you're like, oh, come on. It only took you about a minute to memorize it, so you can worry about other stuff. They're just trying to memorize that stuff. That's what mnemonics do, make your life easier. Okay? Not that you, I figure everybody here has a social life, so I did that for you. Okay? All right, so it's just showing you what this is over here. Okay? All right, and again, this is a sodium-potassium pump. If you know the song, you'll understand how this picture works. Okay? And a proton pump, we'll get into that, making ATP and electron transport chains. We'll talk about those later on. Okay, this is in plants and fungi and bacteria. All right, now we also have something called secondary active transport. This active transport, because it's still going from a low concentration to a high. However, it's not using ATP as its energy. It's using some other energy source, not ATP. So movement against the concentration gradient is going to go from a low concentration to a high. The energy comes from some other molecule. Okay? Some other molecule. Think of a water mill or a water wheel or a windmill. You don't know how these work. I'm going to give you a very brief explanation of how this works. But it's important to understand what this is. It took me a while to figure out this analogy, but it works. Here you have a house. And the house needs to light up. It needs energy. Well, the house, let's say Hurricane Sandy happened and there's no power going to the house. So we have a generator that creates electricity. So there's going to be someplace over here, this generator. And this generator will create electricity as long as we can actually turn it. Okay? Now, a regular generator will have gasoline being able to turn that motor. If you don't have gasoline, you can crank it the old-fashioned way and keep on doing that and make your arms too tired. I do that for a couple days. Or we can have a water mill, or a water wheel rather. And a water wheel is going to use water coming down here and the power of the water is going to turn this. Does that make sense? Right? Well, that's turning, and we can actually connect this to that. We're using the power of water to be turning the generator to be turning on the light. Does that make sense? We're using some other source to make our light turn on. Windmills do the same thing too. My idea, in not the Midwest, but where all the fields are, a lot of wind is over there. Just put acres and acres of windmills and have it connected to the rest of the United States. You're using the power of wind to light up the United States. Okay? Does that make sense? Now, what's happening here with secondary active transport, sodium is the most common driving force that other molecules can get in and out of the cell. Sodium is actually doing this. And we have two different types of secondary active transport. We could have co-transport, also known as synport, and that's where the sodium molecule and this other molecule is going the same way. The best way I can explain it is that, we'll do it this way. I'm going outside. I'm going to go through this doorway. All right? And someone else needs to go out there too. We're both going the same way. But this person, the student, is not going to use energy to open the door. I'll open the door. You're using the power of me that's going to open the door to allow both of us to go out. Does that make sense? That's co-transport, where the sodium, myself, and this other character is going to go out at the same time. But it's under my expense of using my energy to do that. Is that clear? Then we also have counter-transport, also known as antiport. Again, you're using someone else's energy to open that door, but they're going in opposite ways. In other words, I see someone trying to come in here. So I'm not going to allow him to use the energy. I'm the sodium. I'll open up the door. I'm going out. He's coming in. Does that make sense? Is that clear? Okay. So that's the difference between those two kinds of secondary active transports. Keep in mind, the primary active transport is going from a low concentration to a high, but it uses ATP. This one is secondary active transport, where it's going from a low concentration to a high, but it's using some other form of energy that's not ATP. Usually some substance. In most cases, it's usually sodium, at least in the human body it is. All right? So here is, this is going in the same direction. This is co-transport. And one's going one way, the other one's going the other way. That's going to be antiport. right? Questions thus far?
Small molecules, such as sugars and amino acids, can be transported up a concentration gradient. The sugar moves via a membrane transport protein from outside of the cell where the sugar concentration is low to the inside of the cell where the sugar concentration is high. The transport of the sugar through a coupled transport protein is driven by the movement of counterions such as sodium ions or protons that are moving down their concentration gradients from a region of high to low concentration. Sodium ions and the specific sugar or amino acid simultaneously bind to the same transmembrane protein on the outside of the cell, called a symport. When the counterion is sodium, the low concentration of sodium on the inside of the cell required to transport the sugar is maintained by the sodium-potassium pump, which is powered by ATP. In a related process called countertransport, the inward movement of sodium ions is coupled with the outer movement of another substance such as calcium ions. As in cotransport, the sodium ions and the other substance bind to the same transport protein called the antiport, but in this case they bind on opposite sides of the membrane and are moved in opposite directions. The low internal sodium ion concentration is maintained by the sodium-potassium pump.
Alright, it's just showing the same thing. The waiter going in here, co-transport. So the waiter's going in, and I guess these molecules, or no, sorry, these molecules are going in. Or if they go in opposite directions. If you understand that, then these other pictures, they're just falling. Like I said, that's how I study. If I think I understand it by reading a certain way, then I got to look at 2 or 3 or 4 other ways that other people explain it. It's the same thing. But I want to see how other people are explaining it to reinforce the idea in my head. That's how I got through the end part of college, because that didn't hit me until like junior year, and all through medical school. Does it take extra time? Yes. But it's active learning, okay? Not just reading it once and thinking you know it, okay? And here's co-transport. We can see another example over here. All right? This is just a couple of miscellaneous transport mechanisms, because these are words that we're going to be using throughout the rest of biology. Endocytosis and exocytosis. All right? Endocytosis is anything that's going to engulf something and bringing it inside the body. All right? If something's going to engulf a liquid, we call it pinocytosis, like a water droplet or something. If it's going to engulf some microorganisms, something solid, then we call it phagocytosis. Okay? And these, important, these require energy. They use ATP to do that. Okay? So phagocytosis, here's the cell, and you see the solid particle here. It starts absorbing it and getting it inside there, right? That's phagocytosis. All right? And it's kind of showing on a molecular level what that's actually doing. All right? Exocytosis is the opposite. A vesicle from inside the cell, a certain protein needs to go from inside the cell to outside the cell. So it kind of spits it out. So we call it exocytosis. This also requires ATP. Make a list of which ones require ATP, which ones don't, which ones require energy but not from ATP. What does it do? Which way does it go? All right. I can see many different ways that you could come up with charts. Again, I don't study from charts. I think it's bad. But you drawing up a chart is a totally different thing. That's basically trying to organize things on a piece of paper so that you can organize things in your brain. And if you haven't done it before, you keep on doing it and you'll be able to get through college and wherever you're going to go because you're really wiring your brain a certain way. All right? And again, these green balls need to go out, so here's inside the cell. It kind of, the cell membrane kind of fuses with the wall of the vacuole and kind of puts it out, or vesicle rather, not vacuole. All right? Questions?
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins, from the intracellular fluid to the extracellular fluid, using vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. Cells use all these processes, passive and active transport, exocytosis and endocytosis, to move materials across their membranes.